So we're in the process of talking about the process of infection. What happens, or what is the criteria for having a state of infection? You have to be sick? Yeah, organisms have to be growing in tissues that are normally free of microbial growth. And what's the first step that initiates the whole process of infection? Pardon? Yeah, it happens at a portal of entry, and there has to be attachment at that particular portal of entry. So no attachment, no infection. Well, when we talked about microbial structures several weeks ago at the beginning of the class, we talked about some of the structures that bacteria have that will facilitate their attachment to other cells or to each other. And we talked about fimbriae, we talked about capsules. In virus, there are these little proteins poking out from the envelope that are referred to as spikes. But basically, these particular molecules are ligands, and these ligands, these molecules sticking off the surface of bacteria, have to be able to bond to complementary receptors on the cells that they are going to infect. Those particular ligands are oftentimes generically referred to as adhesins. So where might you find adhesins on a bacterial cell? Maybe they're part of the cell wall. Maybe they're part of the pilot or fimbriae. Maybe they're part of the flagella. But there's going to be some molecule on the surface of that bacterium that's going to match up with a receptor site on a susceptible cell that they can attach to. And there is specificity. Okay? That's why, for example, Neisseria gonorrhea, which is the gram-negative cocci that causes gonorrhea, its adhesins are specific for molecules that are only found in the urethra and the vagina. And that's why those are sexually transmitted disease. They don't match up and bind to epithelial mucosa cells, for example. Bacterial adhesins can change as a result of evolution and natural selection. And sometimes organisms can, quote unquote, acquire or evolve new adhesins, which will do what? That'll make them able to then infect different categories of tissues that they couldn't attach to before. Sometimes through mutation, a bacteria will lose an adhesin. It will no longer produce it. In that particular situation, that mutation will then turn a virulent strain of bacteria, a highly infective one, into an avirulent strain. Back in cell biology, and I think Dr. Morgan McCombs used the example in genetics too, Streptococcus pneumoniae and the transformation studies that Griffith did. Remember, there were two strains. There was a virulent strain and an avirulent strain. The virulent strain had a capsule. The avirulent strain was a mutation that had lost the ability to produce a capsule, and the capsule was where the adhesins were located. Okay? It also protected the cells from phagocytosis. But you got to latch on. You've got to be able to gain a foothold, or you can infect the tissues. Let's back up for a second. Since we're going to be talking about things getting established in the tissues, what are tissues? You better be able to respond, or Dr. Rhodes will tell you, right? We've all either had A and P or animal phys, right? What's tissue?
Just cells working together.
Okay, a group of cells that are similar in either structure or function. Okay? And what's another criteria for being a tissue besides the cells having its commonality? They're connected, right? So it's a group of similar cells that have similarity in structure or function. That idea of connectedness is going to be important. What are the 4 types of primary tissues in the human body? 4 kind of categories or classes of tissues. Muscle? Pardon? Nervous? Epithelial? And what's the last one? Connective tissue. Okay? Muscular, nervous, epithelial, connective. The 2 types of tissues that are going to be most relevant to our discussions of infection will be epithelial tissue and connective tissue. We'll talk a little bit about nervous tissue and muscle tissue, but epithelial and connective really provide a lot of the barriers which are the first line of defense against infection. They're both important in your skin, for example, which is your very important outer covering, which, if it's not broken, is really pretty effective in terms of keeping pathogens out. If we look at epithelial tissue, it's typically subdivided into 2 categories or 2 types of epithelial tissue. There is keratinized epithelium. You know what the other one is? Epithelial mucosa tissue. Where do we find keratinized epithelium?
Outer layer of skin.
Yeah, that's our outer layer of our skin. What's keratin? It's protein. It is a tough fibrous protein, and it becomes deposited in skin cells as they move toward the surface, and filling those skin cells up with keratin helps to make it more plastic. It helps to make it more have structural integrity. It really helps to kind of reinforce those cells to be a tougher barrier. Where else do you find keratin in your body besides skin? Hair, nails. Okay, so it's that tough and soluble protein. What's epithelial mucosa? Okay, epithelial mucosa cells secrete mucus. Okay, sometimes referred to as your mucous membranes. Where do you find them? Yeah, anything that is really kind of outside that is inside your body. Okay, the tracts and the ducts in your body. So the digestive tract, the respiratory tract. They're inside your body, but technically they're outside because they're exposed to the outside surface. Okay, lining of the reproductive tract is also mucosa. Why are those cells adapted to secrete mucus? Yeah, good. Mucus helps to coat cells and cover up adhesins, for example, and prevent binding of those cells to the target tissue, the epithelial tissue. If you were an aficionado of the old Ghostbusters movies, it slimes the bacteria, okay, and prevents attachment. And what's your body's response to infection typically? Yeah, increasing the output of mucus. You start producing all this nasty mucus because the body's responding to infectious agents by producing more of this protective substance. Okay? The runny nose is also part of another response we'll talk about later. When you get a cold, you also have an inflammatory response that's initiated, and the runny nose and things is also a result of that. Some epithelial mucosa also is ciliated. You know where you have ciliated epithelial mucosa? You have peristalsis in the intestines, okay? The rhythmic contraction of the muscles would help to move things through. Fallopian tubes. Respiratory tract, your trachea has ciliated epithelium. And what does that cilia do? Yeah, the stuff that's coated by the mucus, then if you don't want it to settle down into your lungs, that cilia beats back and forth and creates kind of a sweeping motion that kind of pushes things back up your trachea and into your oral cavity, so you can cough it out or spit it out or whatever. Okay, so it's kind of a sweeping action. It's sometimes referred to as the ciliary escalator. Okay, and those cilia, as they beat, kind of push things back up and prevent them from getting more down into the deeper tissues, like the bronchi. You know what effect cigarette smoking has on cilia? Yeah, it kind of paralyzes ciliary movement. And that's one of the reasons why smokers are more prone to lung infections than non-smokers, because that part of their defense mechanism has been compromised. So, some pretty good adaptations associated with epithelial tissue for providing a primary level of defense against infection, and particularly from those organisms being able to gain a foothold. Connectives also going to be important as well. What does connective tissue do? Yeah, binds other tissues together, right? Okay. What characterizes connective tissue? How's it different from other types of tissue? Okay, there are cells in connective tissue, obviously, but oftentimes they're not necessarily connected directly to another cell of the same type, like happens in epithelial tissue. The cells are actually embedded in a non-living matrix. And that matrix might be collagen, okay, a secreted protein. Or it might be bone. Okay, bone is connective tissue. If the matrix is solid, then the connective tissue is typically called typical connective tissue. Okay, so ligaments, tendons, bone. But there is an important atypical connective tissue that we're going to be very interested in. And there, the matrix is liquid. What's the atypical connective tissue? Blood. Okay. Blood has cells floating around in a watery matrix. What are the three types of blood cells? Erythrocytes, red blood cells, leukocytes, which are the white blood cells, and we're going to talk about all the different kind of white blood cells when we start digging into immunity. And what's the third type? Platelets. They're actually cell fragments that come from the megakaryocytes. Okay. So we're going to be very interested in atypical connective tissue when we start dealing into questions of body defense systems, because they play an incredibly important role in second and third levels of our immune system. Okay, so let's take a look now at some of those properties that pathogens have that allow them to become established in the tissue and cause infection, and then usually cause disease. So let's take a look at some of the specific virulence factors. These are biochemical molecules. Typically, we break virulence factors down into three categories. One category are the toxins. Another category are exoenzymes. And the third category are antiphagocytic factors. Remember that if there are particular diseases symptoms associated with the infection, it's typically the result of the accumulation of these biochemicals, or these virulence factors, and what effect they have on the cells of the host. However, there is another source of chemicals that can also produce symptoms associated with disease. Various immune system molecules that you produce in response to infection can also give you various specific disease symptoms. This particular situation where products of your immune system cause disease symptoms is called immunopathogenesis. Immunopathogenesis, or sometimes referred to as being friendly fire. One of the things we're going to see later on is that following infection, usually the body responds with a fairly complex response called inflammation. And these inflammatory molecules are oftentimes responsible for producing some of the most recognizable symptoms associated with particular disease, including your watery eyes and your runny nose when you have a cold or fever. Another source of friendly fire are a group of molecules called interleukins. These are molecules that certain white blood cells produce that allow the immune cells to communicate with one another. And they're very important. But when you start stepping up interleukin production, sometimes there are symptoms associated with that. You all know when you're starting to feel off, don't you? And you're starting to feel like you're coming down with something. It's called the prodromium. We'll introduce that term again later on. But you just feel off, you feel kind of, a lot of those initial symptoms associated with that kind of pre-feeling of being sick are the result of these interleukin molecules. There's been some real advancements in using interleukins to try to enhance your immune system to treat chronic diseases like cancer. And they've been quite effective. But large doses of interleukins can have some nasty side effects. And so sometimes these particular treatments give you similar type of responses that other types of chemotherapy would give you for treating cancer. It's kind of a double-edged sword. But ultimately, the negative aspects associated with the production of these molecules is outweighed by the positive aspects of dealing with the infectious agent. Let's start out by looking at this particular category of virulence factors, the exoenzymes. What does that term imply? What's happening to these particular enzymes? They're being secreted. Okay, so these are secreted enzymes. Actually, in lab on Friday, when you evaluate your DNA plates, your starch plates, you are looking for the ability of your microorganism to produce exoenzymes, like amylase, which can degrade starch, or protease, which can break down the milk protein, or DNase, which can break down the DNA. or lipase. Okay? Those are digestive enzymes. The bacteria is secreting them into the environment to break down the complex molecules that surround them. Why are they doing that? Why is it important to have some exoenzymes? Yeah, I mean, those molecules, those macromolecules, starch, DNA, are too big to cross the cell membrane and to get into the cell to provide nutrition. So if you can extracellularly hydrolyze those into amino acids or glycerol or fatty acids, then you can transport those into the cell and use them as a source of nutrition. Well, what's probably going to be the result of many of the exoenzymes secreted by pathogens? What are they going to do? Yeah, they're going to break down defenses. Okay. Many of them interact with defense mechanisms associated with your skin, for example. And they cause the skin to start to erode and develop lesions. And then that enhances their ability of their offspring cells to penetrate through those breaks in the barrier of defense. So, for example, one of the exoenzymes that pathogens can produce is an enzyme called hyaluronidase. And specifically, this enzyme targets a molecule called hyaluronic acid. And this is a very important compound in the intracellular cement, if you like, that joins one cell to another in a tissue. Okay, because remember, the skin is a group of connected cells. So if you break down those connections, the skin starts to fall apart. It loses its integrity as a barrier. Staph and strep, particularly, are notorious for producing hyaluronidase. And one of the things that we're going to see is that some of the worst, most virulent pathogens don't just produce one virulence factor. They might produce a whole battery of exoenzymes. They might also produce some toxins. They might have some antiphagocytic factors. As a matter of fact, after we go through those, we'll kind of talk about a couple of examples, and staph and strep are our main examples of pathogens that produce multiple virulence factors. Sometimes this hyaluronidase is also referred to as spreading factor. It allows the infection to spread by breaking down the integrity of the connected tissue. Keratinase should probably be fairly self-explanatory. It degrades the keratin, that tough and soluble protein that kind of reinforces skin. Collagenase breaks down collagen. Again, starts having connective tissues fall apart. An interesting exoenzyme is the exoenzyme lecithinase. Do we know what lecithin is? It is in some food products. It's actually used as an emulsifier in some food products. Lecithin is also phosphatidylcholine. Do we know what phosphatidylcholine is? It's a phospholipid. Where do you find phospholipids? Cell membranes. Okay. Cell membranes are phospholipid bilayers. So lecithinase basically can cause membranes to kind of start disintegrating. And when cells die because they're lysing, one of the things that typically happen is that lysosomes start to rupture. What do lysosomes contain? Yeah, they contain degradative enzymes, DNases, proteases, etc. And when those start being secreted out into the environment, what does that do? That can potentially degrade other cells in the surrounding environment. So these can then kill adjacent cells.
Is that true for any type of cell, whether it's mutated or normal?
Mutated or normal in what respect?
Like if the cell's cancerous
Mm.
for instance, and you expose it to this, would it lyse regardless, or would it not react?
I mean, probably. But the problem is that means you don't really, if the cancer cell responds the way the normal tissue does, then there's not that selectivity. So it's going to harm a lot of normal cells as well.
Yeah.
Okay. Once this kind of progressive cell death starts occurring, oftentimes initiated by this particular exoenzyme, dead cells tend to form anaerobic pockets. Or abscesses. And in these anaerobic pockets, anaerobes can then thrive, and there are some really nasty anaerobes that produce toxins which are deadly. We're going to talk about both the botulism toxin and the tetanus toxin, and both of those are produced by anaerobic organisms. But lecithinase is kind of an important part of the arsenal of these pathogens. You get them into the tissue, and then they start degrading cells and creating this little anaerobic pocket, basically creating an environment in the body where they can then grow and thrive. So here's an example of some bacteria out here in intact tissue, and the destruction of the intercellular cement that holds those particular cells together, allowing penetration of the bacteria through that barrier, if this were the skin, for example. These are just some examples. I mentioned Heliobacter pylori. Do you remember what Heliobacter pylori is responsible for? Stomach ulcers. Okay. We used to think that stress and diet caused stomach ulcers. Now it's pretty definitively bacteria. One of the things that Heliobacter pylori has is it has some exoenzymes that allow it to burrow through, basically, the protective mucus coating that lines the walls of the stomach. And once it burrows down into the mucus coating, then it can start digesting away epithelial tissue and can cause ulcerations or lesions. These particular bacteria also produce the exoenzyme urease. You know why you produce urease? We're going to do a urease test later on. Urea is an organic molecule that contains an amino group. And urease basically cleaves off that amino group, and the ammonia that's produced raises the pH. That's how we detect the test, is we have a pH indicator. Changes color. So why might it be helpful for Heliobacter pylori to produce urease? Yeah, counteract the stomach acid, okay? Particularly when you're trying to get down into this thicker, mucus layer right here. Once you get down into this mucus layer, you're protected more from the acid. But initially, colonization and trying to get through that mucus layer, producing that urease is really helpful. Okay, this is what I wanted to show you. Kind of a little case study, if you like. Whoops. So what we got going on here? Something pretty nasty, right? This is gangrenous tissue. So I promised I'd leave you a few minutes. This is where we'll pick up next time, and we'll talk about gangrene as kind of a case study of the effect that exoenzymes can have on the body and progression of disease. There are a couple of different kinds of gangrene. There's dry gangrene, and there's wet or gas gangrene. So we'll talk about those and the involvement of exoenzymes. So what I'd like you to do in the remaining 10, 12 minutes is pull your PSI tubes out, both your known organisms and your unknown. Particularly with your unknown, make sure you interpret the results of that, particularly if you've got a gram-negative unknown. And then if you don't have time to take pictures today, make sure you stick those in the refrigerator so that they don't continue to grow and change the results. Okay?
If we fail and have positive and it still reacts, does that mean something that most likely is gram-negative?
No. No, it'll give you results. If they just don't match up with the ones that are described in the question. Okay?
Oh, yeah.
So if you verified your unknown with me,
Right.
it's good. It's good. Sure, you come in. You need to come in today. You don't have time right now. You need to come in today and do this only.
Oh, Jacob, come on, tell me why. Oh, we three.
Like a couple.
But
Good.
it stopped like yesterday. It started getting better, so I'm wondering if it was like towards like the end of it.
If you want to throw this in the refrigerator, I mean, if you're just curious, you might take like this colony and just throw it in an MSA plate and see see what it is. I like their TSIs today.